All right, Class 11, let's get right into it. Today, we're exploring the fascinating microscopic drama of cell division that literally creates genetic variation. Welcome to today's explainer, where we're going to unlock exactly how nature ensures we aren't all just identical clones of each other, all while maintaining a really delicate mathematical balance.
So I want to hook you immediately with this biological dilemma. Think back to your class 10 biology for a second. We know that to create fast genetic variation, nature decided to fuse two cells together. But here is the massive math problem. In humans, a normal cell has 46 chromosomes. If a male cell with 46 chromosomes fused with a female cell with 46 chromosomes, the next generation would have an unsustainable 92 chromosomes. The generation after that, 184. It would literally be a chromosome explosion. So how on earth does nature prevent this from happening?
Well, the brilliant workaround is meiosis. This is a specialized kind of cell division that acts like nature's ultimate biological quality control. Its entire purpose is to reduce the chromosome number exactly by half. We call this reduction division. Now, because it's so specialized, this doesn't just happen anywhere in your body. You definitely don't want the skin cells on your arm having their DNA. This only happens in specialized locations meant for sexual reproduction, like the testes or the ovaries.
Here is our roadmap for today's explainer. First, the problem of doubling. Second, enter meiosis, reduction division. Third, zygotene and the pairing, or synapsis. Fourth, pachytene and its crossing over secrets. Fifth, separation in diplotin and diakinesis. And sixth, anaphase 1, the final split.
All right, part 1, the problem of doubling.
You know, before meiosis evolved, simple organisms just relied on copying mistakes in their DNA, mutations, to create variation. But man, that was incredibly slow. To speed things up for evolution, fusing two entirely different cells together was an absolute game changer. But as we just established, you can't just keep adding 46 and 46. Nature needed a brilliant way to essentially cut the deck of genetic cards perfectly in half before fusing them together. Which brings us to part 2. Enter meiosis, reduction division.
Let's look at the overarching mechanics of how this biological safeguard actually works. The absolute most crucial thing to remember is the number 1. In meiosis, the cell is going to divide twice. We call them meiosis 1 and meiosis 2. But the DNA, it only copies itself exactly one time. That's the secret, right there. You copy the DNA once, but you split the cell twice. By the end of it, you've got 4 cells, and each one has precisely half the original amount of DNA.
Now, the most complex, dramatic, and honestly, the most important part of this whole process happens right at the beginning, in prophase 1. It's so detailed that scientists actually break it down into these 5 mini-steps. It kicks off with leptotin, where the DNA kind of just compacts and packs its bags, becoming visible under a microscope as these long, thin threads. But the real magic of genetics, that kicks off in the very next step.
Step 3, zygotene, the pairing, also known as synapsis.
Okay, I want you to picture a massively messy room with a giant pile of shoes. You've got 46 shoes in total. 23 came from your mom, 23 from your dad. For every left sneaker, there's an exact matching right sneaker of the same brand and size somewhere in that pile. In zygotene, the cell has to dig through and find those matching partners, the homologous chromosomes. This pairing process is called synapsis, and just like literally tying the shoelaces together so you don't lose the pair, the cell glues these matching chromosomes together using a crazy little protein structure called the synaptonemal complex. And get this, it's so incredibly tiny, you literally need an electron microscope to even see it.
Let's nail down this Class 11 vocabulary real quick so we're perfectly clear. When those two matching homologous chromosomes lock together, the whole pair is called a bivalent, because, there are 2 chromosomes. But remember, the DNA already copied itself, right? So each chromosome actually has 2 arms, chromatids. That means in one bivalent, you have 4 arms total, which we call a tetrad. So if a human cell starts with 46 chromosomes, in zygotene, it forms exactly 23 bivalents. Boom, math checks out.
Moving right along to section 4, pachytene and crossing over secrets. All right, I'm going to slow the pacing down for a second here because this is it. This is the absolute most crucial stage of today's entire explainer, pachytene. If you understand pachytene, you understand why you look a little bit like your parents, but not exactly like them. Before we look at the swap, you got to know who is doing the swapping. Take a look at this distinction. Sister chromatids are the 2 arms on the exact same chromosome. They are identical clones of each other. If they trade pieces, nothing changes. But non-sister chromatids, those are the arms belonging to the different chromosomes in that glued-together pair we just made. One from mom, one from dad. The magic of crossing over only happens between these non-sister chromatids.
So how does this crossing over actually work? Well, those non-sister chromatids wrap around each other super tightly. And right where they touch, this thing called a recombination nodule forms. Think of it like a little molecular workstation. Then, a highly specific enzyme called recombinase steps in. It acts like microscopic scissors and tape, literally cutting a piece off mom's chromosome, cutting a piece off dad's, and swapping them. This right here is crossing over. It is the ultimate source of genetic variation in the universe. It's exactly why every single sperm or egg cell you will ever make is totally 100% unique.
All right, Section 5, separation. Let's look at diplotin and diakinesis. So the genetic secrets are swapped. Now that synoctonymal complex, that protein glue holding the pairs together, it needs to dissolve. This happens in diplotin. The chromosomes try to pull apart, but they kinda can't. Not completely anyway. They actually get physically stuck at the exact spots where they just traded DNA. Under a microscope, these stuck points look like tiny little X marks, and we call them chiasmata. Now here is a totally mind-blowing fact that you'll definitely see on your NEAT exams regarding this stage. In human females, this exact diplotin stage, it's put on pause before a baby girl is even born. The cells literally arrest in this X-shaped holding pattern and just wait, sometimes for 13 or 14 years until puberty, before they finally continue cell division. Nature literally hits the pause button for over a decade. Crazy, right?
Okay, cleanup time. This is diakinesis. Those X-shaped chiasmata, they slip all the way down to the very ends of the chromosomes, a process we call terminalization. The chromosomes finish condensing so they're nice and thick and visible. The cellular machinery, like spindle fibers, forms up, and the nuclear envelope completely breaks down. And just like that, prophase I is officially a wrap, and the cell is fully prepped for the main event. Which brings us to our final section, part 6, anaphase 1, the final split. So we're going to fast forward just a bit past metaphase 1, where the pairs simply line up on a double plate in the middle and go straight into the high-stakes separation, anaphase 1. Now, in normal cell division, chromosomes are ripped right down the middle. But actually, scratch that, not here. In anaphase 1, the entire homologous chromosome, with its brand new genetic variations from crossing over, gets pulled whole to one side of the cell, while its partner gets dragged to the other. The sister chromatids, they stay perfectly attached. Because entire whole chromosomes are moving away from each other, this is the exact specific moment the math changes. The chromosome number drops from 46 to 23. This big separation is called disjunction. But, nature isn't always perfectly precise. To wrap up today's explainer, I want to leave you with a bit of a provocative question. What if the glue doesn't unstick? What happens if an entire pair of chromosomes accidentally gets dragged to one side of the cell without separating at all? Well, this failure is called non-disjunction. It results in one cell getting 24 chromosomes while the other only gets 22. And when these eventually fuse later on, it leads to conditions known as aneuploidy, which is actually the root of a lot of genetic disorders we'll be exploring in our future lessons. It really makes you think about how incredibly delicate this microscopic dance truly is. Keep studying Class 11, and I'll catch you in the next explainer.